Hello, welcome to Science Breakthroughs from Science Mission. I'm Sadashivapai and going to discuss a couple of breakthrough papers published in the week of July 28th. Let's start. First, we will discuss a paper published by Paolo and colleagues in Cell Reports on the identification of a mitochondrial switch to control tumor metastasis. Why is this paper so important? The role of aerobic glycolysis in tumor metastasis is well known. Authors identified a different switch primarily affecting mitochondria. Mitochondrial electron transport chain overload and inhibition forms a switch with superoxide formation in both the cases. Authors also identified downstream effectors of the switch. Let's look at the data. From wild-type population of SIHA human cervix carcinoma cells, authors isolated F3 cells following 3 rounds of in vitro selection as shown here. These F3 cells had enhanced invasive activity, increased migratory phenotype, enhanced glucose uptake, and surprisingly, no higher lactate production. They also had increased mitochondrial oxygen consumption and succinate accumulation. When authors subjected moderately metastatic F10 cells to several rounds of in vivo selection, as shown here, they found increased metastatic activity, increased invasiveness, and clonogenicity. However, there was no difference in the rate of glucose uptake and lactate release and mitochondrial oxygen consumption, but increased succinate levels were seen, indicating TCA cycle overload in the metastatic cells in vivo. Authors generated P0 with damaged mitochondria and P0D2 as represented here with total destruction of mitochondria. Damaged mitochondria, that is P0, when treated with retenon resulted in decreased oxygen consumption, altered metabolism, increased migration, and invasion. However, total loss of mitochondrial function, that is in P0D2 cells, resulted in decreased migration and invasion, indicating partial mitochondrial function is required for metastasis. Partial oxidative phosphorylation inhibition with low dose of retenon targeting complex 1, 3-nitropropionate, complex 2 inhibitor, or a specific siRNA against complex 4 subunit increased migration and was blocked by antioxidant N-acetylcysteine and epsin. Similar result was also observed using mitochondrial oxidative phosphorylation inhibitor MI-tempo. Authors showed SERC, SMAD, and PYK2 pathway mediated mitochondrial ROS tumor metastasis in various in vitro model systems and was blocked by antioxidant and siRNA for SMAD. Metastasis in mice after tailwind injection of F10 tumor cells treated with retenon was abolished by mito-tempo and antioxidant mito-Q. Similarly, IP injections of mito-tempo prevented spontaneous metastasis in M4B mice. These results clearly demonstrate scavenging of mitochondrial ROS prevents progression of tumor metastasis, and this is an important finding. Next, let's look into another paper published by Thomas Trimarchi and colleagues in Cell. Authors demonstrate a role for long non-coding RNAs. I'll be calling them link RNAs in acute leukemia. Why is this paper so important? Role of long non-coding RNAs, that is link RNA, in leukemia is not known. Authors demonstrate Notch1 controls the expression of link RNAs in T-cell acute lymphoblastic leukemia, that is TAL. LUNAR1, a link RNA regulated by Notch1 in leukemia, controls IGFR1 mRNA expression and its signaling. Let's look at the data. Comprehensive mapping of link RNA in TAL showed average link RNA expression was lower than protein coding gene expression. Authors found striking specificity in link RNA expression for TAL and normal T cells. Link RNA promoters displayed higher enrichment of transcription and Notch1 promoter signals. In addition to protein coding genes, many link RNA genes were differentially expressed upon gamma secretase inhibitor treatment that knocks down Notch1 signaling. Approximately 55% of link RNAs whose expression was Notch dependent were directly occupied by Notch1. Authors found LUNAR1, one of the link RNAs widely affected in leukemia. They found LUNAR1 downregulated upon Notch1 inhibition, overexpressed in primary TAL, and expressed significantly higher in TAL samples that harbored Notch mutation, and localized in the nucleus, which is characteristic of link RNAs. Authors developed in vivo xenograph assay in which human TAL cells expressing an shRNA targeting LUNAR1 or Renilla control injected into sublethally irradiated immune deficient mouse, and their immunofluorescence was measured in the peripheral tumors. Authors observed significant loss of representation of cells in which LUNAR1 was depleted, which suggested that link RNA is required for efficient tumor growth both in vitro and in vivo. Ectopic expression of IGF-1R efficiently rescued the growth of defects observed following LUNAR1 depletion in TAL cells, suggesting IGF-1R is indeed a key target of this link RNA. Further, abrogation of LUNAR1 using two independent small hairpin RNAs targeting distinct regions of the transcript led to repression of IGFR1. These results indicate LUNAR1 regulates TAL proliferation by enhancing IGF-1R expression. Authors observed a gene expression assay to determine if link RNA activates gene. Tethering LUNAR1 to this reporter stimulated transcription of the reporter to a similar degree as shown for activator hot tip. Additionally, ASO-mediated depletion of LUNAR1 significantly reduced this stimulatory effect. Using chip assays, authors show that intronic IGF-1R enhancer activates LUNAR1, which then co-occupies the element and further recruits mediator in order to sustain full activation of IGF-1R promoter. A model is shown here. These results clearly demonstrate a role for link RNA in leukemia. Thank you for watching Science Breakthroughs from Science Mission.